Hello, my name is Brendan O'Farrell. I'm the president and founding partner of DCN Diagnostics.
And I'm Pat Vaughan. I'm the Chief Operating Officer here at DCN, and I run all our development programs.
I guess it's useful to ask the question: what's the purpose of doing a diagnostic test? Right. The world right now is very focused on, the lack of testing, or the effectiveness of testing, or the speed of testing, and why the infrastructure hasn't been able to keep up in some places. So I think it's useful to understand the purpose of diagnostic testing, right? So at its simplest, in order for a diagnostic test to be useful, we have to be able to effect an action based on the result, right? There's no point doing the test if we can't do anything with the result. It's also essential that the results be generated in a time frame in which an effective action can be taken. So in the case specifically, I suppose, of COVID, what's important at various points in the pandemic will change, right? What's important at the very early stage is quick identification of positives, and as that matures, particularly quick identification of the asymptomatic or mildly symptomatic cases, so that we can actually help to prevent the exponential spread of this thing. So as time goes on and the population becomes more and more exposed, then some of the testing paradigm changes, and it switches more to the detection of antibodies. And so we can identify people who have been exposed, but perhaps have not developed any symptoms, and we can identify people that have been exposed and recovered. So those testing paradigms are different. The first wave of assays that have been deployed are RT-PCR-based assays looking for antigen, effectively, right? They're looking for the presence of the virus or the molecular product associated with the virus in infected individuals. Moving on from there, then there's an opportunity to move into protein-based testing for the same thing. So we're looking for viral antigens in the bloodstream, or in nasopharyngeal swabs, and again, just looking at the presence of the virus itself. Moving on from there, then we'll move into serology testing, where we're looking for the immune response that says, yes, I've been exposed, I've been perhaps infected, and I've developed an antibody response to the presence of the virus. So those things have different purposes and different values at different points in the pandemic.
Obviously, molecular testing is of the essence today. It's been rapidly deployed. The assays have been developed. But this infection, this pandemic, this COVID-19, that's not going to go away tomorrow either. Okay, so a lot of effort has been putting into flattening that curve. But as time goes on, more people will have been infected, but also generated an immunity. The virus is not suddenly just going to disappear. It's going to be with us, just like flu. Flu comes back every flu season, every winter. A new flu strain comes out and infects people. So while immediate testing with molecular tests is very important, there is a need for these rapidly deployable, easy to use rapid tests for the second wave of infection and for future uses. So, this time next year, okay, what are we going to be testing for as well? It's not suddenly going to disappear. We're still going to be testing for COVID-19. The incidence is going to be a lot lower. The rate of infection is going to be much, much reduced. And that because we will have these rapid tests deployed where we can test and get immediate answers to whether somebody is infected or not. If they're infected, they're immediately quarantined.